Welcome to the second unit of second section of human physiology paper. The topic of today's presentation is blood clotting. When we talk about blood clotting, we should not forget about the scientist Paul Morowitz, because he is the one who identified the pathways that is responsible for blood coagulation. He even wrote a book, Blood Coagulation Pathway. So in that manner, first we should thank about Paul Morowitz for its important contributions towards this blood clotting. Blood coagulation or clotting. So the conversion of the liquid part of the blood, which is converted into a solid, semi-solid like substances with a combination of cells and proteins, is known as the blood coagulation or the blood clotting. So the factors which are going to promote formation of blood coagulation or clotting is known as the procoagulants, and the factor which prevents the formation of this blood clotting is known as anticoagulants. So in a body, both procoagulants and anticoagulants are present. Depending upon their composition, their ratio, it may cause coagulation or it may not cause the coagulation. And moreover, when the role of anticoagulant is high, the blood is flowed through the vessels. When the concentration of procoagulants are increased, then the blood gets clotted. So this is what the basic phenomena of blood clotting. So blood clotting occurs within the blood vessel itself. Sometimes blood clot occurs, and it can occur by means of injury to the blood vessel also. Blood clotting occurs. So in both ways you can expect the blood clotting. So the clotting might occur within the blood vessel, or the clotting might occur when there is a damage occurs to the blood vessel. In general, the blood coagulations occurs in three major steps or stages. The first stage is the activator, formation of a prothrombin activator. When the damage occurs to the blood vessels physically or internally, if the blood vessels damaged in the interior part without causing the damage to the blood vessel, it can also induce the blood coagulation steps. Or when there is a physical injury occurs to the vessel, when there is a leakage of blood occurs, at the time also the first stage, that is what formation of prothrombin activator occurs. The second stage, the prothrombin activator converts prothrombin to thrombin. Then the third step is the conversion of fibrinogen into fibrin by the action of this thrombin. Once the fibrinogen is converted into a fibrin, and then it gets polymerized, and then it forms a crisscross structure, and it prevents the movement of the cells, that leads to the formation of a clot. So clot is going to nothing but the en-masse platelet, blood cells and plasma together, which prevents the loss of blood. That is known as a blood coagulation or blood clot. So this is an overview about the blood clotting process. As I told, the first factor to be formed is prothrombin activator, and then this prothrombin activator converts prothrombin to thrombin. Then the thrombin is going to convert fibrinogen to fibrin monomer, and these monomer gets polymerized, and then these polymerase are cross-linked, and these reaction is also catalyzed by this thrombin. So these are the three stages. Prothrombin activator is the first stage, thrombin formation or activation of prothrombin is the second stage, and conversion of fibrinogen to fibrin is the third stage. So these are the three stages of blood clotting. And these are the factors which plays a vital role for this blood coagulation. These factors are called as clotting factors, coagulation factors. Factor 1 is fibrinogen, factor 2 is prothrombin, and factor 3 is thromboplastin, factor 4 is calcium, and factor 5 is proaccelerin, or it may be called as labile factor or accelerator globulin, and factor 7 is proconvertin, SVCA and stable factor. Okay, then factor 8 is the antihemophilic factor, antihemophilic factor A or antihemophilic globulin. So different names are given to this factor. Then factor 9, plasma thromboplastic component, Christmas factor, anti-hemophilic factor B, then 10 is the start factor, 11 is the plasma thromboplastin antecedent factor or anti-hemophilic factor C, and then the 12th factor is the Hagman factor or glass factor, and 13 is the fibrin stabilizing factor or Lacky-Lauren factor, and HMWK, that is a one factor which is known as high molecular weight kininogen, and pre-Ka, pre-kallikrein and kallikrein and platelet phospholipid. So up to 1 to 11 you can see these are considered to be as main important factors of blood coagulation. You can see that there is no sixth factor is present in the blood coagulation. So there is no sixth factor. Only 1, 2, 3, 4, 5, 6 is not there. 7, 8, 9, 10, 11, 12 and 13 is a number. So that is sometimes that is they initially labeled some protein as a factor 6, but later they come to know that the factor is not going to involve in blood clotting, so it is removed from the list. So that is how the factor 6 is not listed in the blood clotting. The number is not given as the order of their function, so it may vary. Sometimes it may say, for example, the end component of the blood clotting is a fibrinogen, so it is given as a number 1. The second was going to be a prothrombin, but afterwards we can't expect the same order the factor to be involved in the blood coagulation. So it may vary. The order of role played by the factors will differ. Okay, then we will go each step. First step is the prothrombin activator formation. So how this prothrombin activator is going to form? The prothrombin activators are formed in through two pathways. Either one pathway is actually occurs at a time. So one pathway is known as the extrinsic pathway, another pathway is known as the intrinsic pathway. The word extrinsic and intrinsic means that whether there is any damage occurs to the blood vessel. If the blood vessel is damaged and blood comes out of the vessel, leakage occurs. When a tissue damage occurs, that case leads to the pathway known as extrinsic pathway. When the damage occurs within the blood vessel, when the blood vessel is injured interior part inside the blood vessel, then it will induce a pathway known as the intrinsic. So intrinsic means that damage is not found to be an extrinsic. Blood is not going to come out of blood vessel. That is the pathway is known as the intrinsic pathway. When the damage, when the injury occurs as an extent of damage of blood vessel, then it leads to the pathway known as extrinsic pathway. So now we will see one by one, but these two pathways plays a vital role in the first step of blood coagulation. Don't forget it. Okay. So extrinsic pathway. As I told earlier, when there is a blood vessels are damaged, when tissues are damaged, the extrinsic pathway for initiating the formation of prothrombin activator begins with a traumatized vascular wall or traumatized extracellular tissue that come into contact with the blood. So blood comes out of blood vessel. In that cases, this extrinsic pathway is stimulated. The first factor which is going to play a vital role in extrinsic pathway is going to be a tissue thromboplastin, or it is called as a tissue factor. And this tissue factor is nothing but a lipoprotein complex, which is made up of phospholipids from the membrane and a lipoprotein complex. And when they combine together, they act like a proteolytic enzyme and they activate factor 10. And then the factor 10 is not only activated by tissue factor, it is also required the role of factor 7. Okay, then factor 7 and the tissue factor activates factor 10 to factor 10A. Even calcium ions also required for this activation process. Once this factor 10 is converted into 10A, you can see here, once the 10 is converted into 10A, by this tissue factor and factor 7 and calcium ions, they can combine with calcium ions and tissue factor and one more factor, that is factor 5. So factor 5, calcium ions, tissue factors and activated 10, that is 10A, will combine to form a complex which is known as prothrombin activator. So now the prothrombin activator is ready. That is the first stage of blood clotting completed through extrinsic pathway. When there is a damage to the tissue occurs, when the blood comes out of the blood vessels. So the tissue factor is going to be a starting factor, and then it will activate factor 7, and then factor 10, and then factor 10 along with other components like factor 5, calcium ion, tissue factor. They will form a complex known as a prothrombin activator. Then the remaining steps is going to occur the later time. Okay. Then in intrinsic pathway, the second mechanism for initiating formation of prothrombin activator and therefore for initiating clotting begins with trauma to the blood or exposure of blood to the collagen from a traumatized blood vessel wall. So when the interior part of the blood vessel is damaged, then the cells which are running and the proteins which are present inside the blood vessel can interact with the collagen. Where the collagen is present, it is in the connective tissue. So in the connective tissue, which is found to be as a layer of the blood vessel wall, it will interact. Okay, so when the blood cells or the blood is in contact with this collagen, that is the signal is produced to stimulate the intrinsic pathway. So the intrinsic pathway is started by the blood trauma or contact with the collagen. So this reaction is actually activates factor 12. When the factor 12 is converted into activated 12A, the activated 12A along with this high molecular weight kininogen and precallikrin will in turn activate the factor 11. Once the factor 11 is activated, and these factor 11 is again along with calcium ion will activate factor 9. So 12, 11 and 9. Once the 9 is activated in the presence of 8, the 8 is also activated, and along with calcium ion, along with this factor 9, it activates factor 10 to factor 10A. Once the factor 10A is formed, this factor 10A along with platelet phospholipid and calcium ion and factor 5. So this remains similar to the previous intrinsic path, that is extrinsic pathway. So the calcium and then the factor 5 and the factor 10A, that is activated 10A along with this platelets. In the case of extrinsic pathway, it is going to be a tissue factor, but here instead of tissue factor you are going to have platelet phospholipid. Okay, why you are getting platelet phospholipid? Because platelets might be get damaged during the process of this blood trauma. So that phospholipid along with this calcium ion along with this factor 5 and the activated 10A will form the prothrombin activator complex. Again the remaining steps are going to be same. So this is the first stage, activation of prothrombin, formation of prothrombin activator. Then we go to the second stage of blood clotting, that is the conversion of prothrombin to thrombin. You can see this is the polypeptide chain of a prothrombin. It is going to have a chain, a length chain of about 1 to 579 amino acids are going to present in this prothrombin. It has different sites for the proteolytic cleavage. The two major sites are 271, 320, and they also has a site like 284. Okay, then they also has a site for 155. Okay, and then they have 272 site is also going to be there. So it has different proteolytic cleavage sites. So depending upon which proteolytic factor is going to act on this, you will get different polypeptide chains. So usually these are the two important sites which are necessary for converting prothrombin to thrombin. So the reaction is actually carried out by the factor 10A or prothrombin activator complex. So this prothrombin activator complex, if it can cut this 271 point, when it cuts in 271 point, you will get prethrombin 2 and the remnants 1 to 271, PF1. Okay, 1.2 you will get. PF1.2 will get when this factor 10A will cut at 271 and then you will get prethrombin. Then when the factor 10A again cuts at 320, you will get thrombin, which is made up of 272 to 579 amino acids length. Sometime the factor, that is this 320 point is initially cut, so you will get mesothrombin. Then the 271 point is cut, then you will get thrombin. So the two ways the thrombin is produced, but still the factors This thrombin is actually a functional protein which is going to play a vital role in the case of clotting. And this thrombin can also be further degraded. After its function is over, it might be degraded by the action of the other factor that is known as the factor 2A. 2A is nothing but the activated thrombin. So the activated thrombin itself has an ability to break down this thrombin to form a 280-320, that is 284 point. The length is going to be 272, no, 272 to 284 amino acid is removed by the action of again the same thrombin to form a alpha thrombin. And there is also different ways in which these 2A factor and this 10A can also break down this thrombin. So this pre-thrombin 1, if it is cut in this 155 by this 2A, it can convert this pre-thrombin to PF21 prothrombin. So this is also another regulatory mechanisms which are going to decrease the level of thrombin after its activation of clotting, or to control the role of prothrombin from the clotting process. So these regulatory events are going to be carried out by these reactions. Okay. So the main reaction is prothrombin, which is going to cut these 2 points, R271 and 320, to result in the formation of thrombin, which are actively involved in blood coagulation process. That is the stage 2. Then stage 3, that is conversion of fibrinogen into fibrin. The first stage you can see here. This is a fibrin monomer. It is going to contain 2 domains. One domain is D, another domain is an E. And it is also containing a polypeptide chain known as A and B. Okay, so when thrombin acts on this fibrin monomer, it actually removes this A and B domains. How it removes the A and B domains, that is what the reaction you are seeing here. So thrombin will cut the peptide bond between arginine and glycine of this fibrin monomer, and it releases alpha and beta polypeptide from the thrombin, alpha chain and beta chain. So once this alpha chain and beta chain A and B residues are released from this fibrinogen monomer, then the fibrinogen monomer can get polymerized by interacting with another monomer. How the monomers will going to interact? The D domain of one fibrin can interact with E domain of another fibrin. How they will interact with each other? What is the type of bond they are going to form? They are going to form a bond like this. Peptide bond is going to form between these lysyl amino acid, lysine residue of one domain, and to the glutamyl residue amino acids of the other domain, to form a peptide bond by the action of the enzyme transglutaminase. That is nothing but a factor 13A. So the factor 13A will form the bond between E domain and D domain of one fibrinogen to the other fibrinogen. So that is how the fibrins are cross-linked, crisscross. It can cross at any directions. It can interact with that E domain, with the D domain, D domain. So another E domain, D domain. So it will form a crisscross fibers. And this crisscross fibers, which are going to present in between this D domain and E domain, will make the fibrin polymer formation. Once the clotting occurs and the blood stoppage, leakage of blood is stopped, you have to dissolve the clotted material, and this reaction is actually carried out by the plasmin. So that is what the last step. So plasmin can degrade, break these D and E domain interactions, and then they will dissolve the clot. So that is carried out by the plasminogen at the fibrin degradation stage, fibrinolytic system. So that is actually a different system. It is not a single step, it's a multi-step process which will discuss in a blood clot lytic mechanisms. So up to the formation of a fibrin polymer is the clotting process. This is the third stage of blood clotting. Now you can see the image where the fibrin crisscrossed fibers blocks the RBC and other blood cells so that their cells are not moved. So this is how the clot looks in an electron microscope. If you see an electron microscope, the RBC gets trapped in between the fibrin fibers. Because of this trapping, it won't allow the leakage of blood at the place of damage, injury to the blood vessel. So that is how the clotting prevents the loss of blood Okay, so this is how the clotting plays a vital role. So this diagram once again shows the complete mechanisms of this blood clotting. So the clotting occurs in three stages. One is the formation of prothrombin activator. It may be formed in extrinsic pathway or it may be formed in intrinsic pathway. In both cases, you are getting a complex which can activate this 10. So 10A is considered to be as a prothrombin activator. Once prothrombin activator is activated, the pathway remains common. So how this factor is activated, that is what different in extrinsic and intrinsic pathway. Once the factor activates this 10A along with factor 5 and calcium and phospholipids, it can activate prothrombin to thrombin. Then thrombin converts fibrinogen to fibrin, and fibrin forms a polymer, and this polymer is going to prevent the blood loss. So that is what the ultimate aim of blood clotting. Okay, so the factor is going to involve in extrinsic pathway is mainly factor 7. And you can see in intrinsic pathway, it is going to be 12, 11, 9. In common pathway, 10, 5, 8, 7 by 13 is going to involve in the common pathway, because 8 is going to involve here in the case of intrinsic pathway. So this is how the factors are distributed. And even liver plays a vital role in the blood clotting, because liver is the one which is going to synthesize all the clotting factors. And they also synthesize bile salts through which the important vitamin which is required for the blood coagulation, vitamin K, is going to be absorbed only because of the bile salts produced from the liver, because vitamin K is a fat soluble vitamin. So if you want to absorb any fat soluble substances, you need bile salts. So that bile salt is actually synthesized and produced by liver. So in that way, the liver plays vital role in the blood coagulation. And then the coagulation is prevented by different factors. For example, a tissue factor pathway inhibitor, secrete tissue factor pathway inhibitor, which is going to inhibit the role of 7A. So the formation of prothrombin activator 10A is not going to carry it out. So secrete tissue plasminogen activation factor. So once plasminogen is activated, tissue plasminogen activator catalyzes the formation of plasmin. So once plasmin is formed, the clot is dissolved. So like this, several anticoagulating factors are there. So these anticoagulating factors are produced within our body. Okay, here also you can see protein C. They can also act as an anticoagulation factor. So we have different anticoagulation factor within our body. But we can also use anticoagulation factor externally, artificially. So when you take a blood from a patient, if you want to avoid the blood clotting, you can add anticoagulants. For example, EDTA is used as an anticoagulant. Oxalate, you can use citrate, components are used. Okay, so they are acting as a chelating agents, calcium chelating agents, EDTA. So that is how they prevent. They take up all the calciums from the blood with themselves, so they are not available for clotting process. So clotting is inhibited. Okay, so by using these type of factors, you can prevent clotting in an artificial manner. But inside the blood, the clotting is prevented with the help of these type of anticoagulation factors. Heparin, you may hear of it. It can also act as one of the anticoagulating agents. Okay, then clotting time. So what do you mean by clotting time? So the time required for formation of a clot is known as a clotting time. How you can identify a clotting time? Simply you can withdraw blood and you can place the blood in a test tube, and then you will just move the test tube up and down, and you can see when the movement of the blood stopped. That is known as the clotting time. Usually 6 to 10 minutes is required for a clotting. Bleeding time. So how much time it takes to bleed? If you make a prick at the end of a finger, and then you find out up to what time the blood comes out of that particular tip of the finger, then if you note it out, that time is going to be the bleeding time. When it stops flow of blood, that is known as the bleeding time. So it actually 1 to 6 minutes. It can be tested at the tip of the finger or at the end of the ear lobe. You can test it. Accumulation of blood in the tissues can occur as a result of bleeding from any vessel type, that is known as the hematoma. You can see the red color patches occurs in some part. due to this allergic reaction and all. So that type of things are known as hematoma. Then prothrombin time. So prothrombin time gives an indicator of the concentration of prothrombin. So the prothrombin time is actually calculated to identify the availability of prothrombin present in the blood vessels. So for this, what they will do is they will remove blood removed from the patient immediately, and then it gets added with the oxalate, so that it avoids the conversion of prothrombin to thrombin. Then large amount of calcium ion is added and tissue factor is added quickly with the oxalated blood, so that they will calculate the amount of time taken for the formation of the clotting. So actually the normal prothrombin time is going to be 12 seconds. So when you add more amount of calcium and tissue factor, that means they are not the limiting factor. So the clotting is going to occur. So in that mechanism, which shows the amount of prothrombin. So if you have very less amount of prothrombin, then it indicates the time taken will be more. So if the normal prothrombin time is going to be 12 seconds. Okay, so if it takes longer, it means that the prothrombin level in a person is going to be very less. And then these factors, we saw around 12 factors which are plays vital role in the case of blood coagulation. Of these factors, if you have any one factor is deficient, you might have some diseases. So we know the name of the factor itself, anti-hemophilic factor. So those factors, what it indicates, if the factor is not there, you will have a disease known as hemophilia. Like that you can see the factor 8. If you are not having that factor 8, you will have a disease known as hemophilia A. If you are not having factor 9, you will have a disease of hemophilia B. So like that, the deficiency of each factors will lead to some particular diseases. Even Christmas factor, 1 milligram factor. There are so many factors which will lead to different diseases. You can see here hemophilia B is caused by the loss of this Christmas factor. So that is how loss of clotting factors can lead to different inherited disorders. Okay, so that is why most of the diseases are referred as a congenital, because it's a inherited due to lack of genetic defects. And then the clot, once the clot is formed, the clot has to be dissolved within our body. If the clot is not dissolved, then it can lead to several diseases. Because if the clot is not dissolved properly, the fragmented clots can block the flow of blood in minute vessels. Everybody knows it. If blood doesn't flow to the heart, it will lead to cardiac arrest. If the blood is not moving to the brain, it will cause brain death, coma. So if you want to avoid such defective conditions, we need the clot has to be dissolved once the region is recovered from the damage, or the damage is rectified. Then the clot has to be dissolved. And we have a set of system like this protein C, which is converted into activated protein C. And they can also bind with protein S, and they can inactivate factor 8 and factor 5. We know that factor 5 and 8 is going to play a vital role for the prothrombin activated formation. So they are going to inactivate them. Then there is also the activated protein C, inactivates tissue plasminogen activator. Inactivates inhibitor of tissue activation plasminogen activator. So that means what? When the plasminogen is become activated, become active, then it converts plasminogen to plasmin. Then this plasmin actually break down the fibrin cross layer. Once the cross polymer of a fibrin is broken down, the RBCs are free and they can move away. So the clot is now no more. The clot gets dissolved. That is how this fibrinolytic system functions. So fibrinolytic system is very, very important for our body. If the fibrinolytic system is not functioning, then there may be a blockage of blood occurs even in the brain. Emboli formation in the brain is because of this improper lysis of blood clot which is formed inside the body. Okay, so this is how this fibrinolytic system is very much important as like blood clotting system. Okay, so with this I conclude the second unit of physiology paper. If you have any doubt, you can contact me through phone or mail ID, 9865866168 or sivabio@gmail.com. I will reach you through mail or phone. And the books like Ganong Review of Medical Physiology, Vander's Human Physiology, and Fox Human Physiology, and they are all used for the preparation of these presentations. With this, I conclude this session. Thank you. Thank you very much.